Hello everyone, welcome to another session of Biotechnology. We are delighted to have Bob Steininger, who is the Senior VP, CMC at Surface Oncology with us. Welcome Bob.
Thank you very much.
Bob graduated from MIT in Cambridge in 1976, and then he did his master's from the University of California at Berkeley in 1978.
Correct.
And he has been involved with and associated with biotechnology from the 1980s, where in the early days he was associated with fill, primarily alcohol, generating alcohol through biotechnology. He has been associated with companies like Genetics Institute, Wyeth, American Home Products, Millennium Pharmaceuticals, Acceleron Pharmaceuticals, Voyager Therapeutics, and now Surface Oncology. Bob has been associated with more than 30 product development regimens and cycles, and has been responsible for release of commercial products like EPO, GM-CSF, and Factorate. Welcome, Bob. Welcome to Biotechnology.
Thanks very much.
So it would be nice, Bob, if you could just introduce yourself a little bit from what I've done in terms of just giving our viewers of this channel of biotechnology a little bit perspective about biotech from the 1980s. That would be fantastic.
Sure. So when I started, I was truly a chemical engineer, process engineer, and I focused as a graduate student very much on change of organic material into fuel. But quite frankly, part of that was very applicable to processes in general. And as I worked on processes, certainly when I started in the pharmaceutical industry, quite frankly, they didn't have process engineers.
Sure.
And to a certain extent, that was my role in the first job that I did, is really focusing on, well, how would you make material if we were going to go and make a protein as a product? And what would you need to do to actually do that in a method by which you could make it consistently and actually make sufficient material? And certainly making proteins was not something that most people did.
When we see the journey of yours from 1980s to now, I'm sure there have been fundamental changes that you have seen and you have been associated with. Before we get into the future of process development and manufacturing, could you outline for some of our viewers these sort of changes in hardware or changes in the CMC issues that you've sort of dealt with and seen the changes? It would be wonderful.
Sure. Certainly probably the major change is that we started actually making proteins that in fact were mostly isolated from plasma, from human beings that basically had that protein, but they didn't have very much. And our role originally was to make those proteins in significant amounts so that you could use that as a drug. What we've gone from that to actually making proteins that are novel, different, and have different properties. Many of them are relatively easier to make. If you're talking about antibodies or even some of the fusion proteins you can make. But to a certain extent, that's a different entity unto itself. And there's a lot more homogeneity as far as those proteins compared to what we originally made. And certainly a lot easier way of actually purifying and scaling up those processes correctly.
True, nowadays we get to hear a lot of gram per liter.
We do. I was making less than 10 micrograms per mil in some of the things that we were making.
Right.
So significantly less.
Significantly less. And now, do we deal with problems of plenty now? Do you think the issues of downstream processing has changed from when you came in, or is it primarily the upstream that's gone up?
So right now, quite frankly, we've done extremely well in making more material in the tanks, in the bioreactors, and the fermenters. The reality is processing that in the purification side and actually making the material for a drug product is limiting, particularly the purification, particularly making that as far as proteins. And to be able to do that in a way that is less expensive and more efficient is certainly an area that we need to actually think more about and actually focus upon. That is the most expensive part of making proteins for most of the highly expressed proteins that we make now.
Absolutely. So when you now compare your journey to what you've done from 80s till now, how do you see the next 20 years for the future of process development and manufacturing? What do you see are the new trends that are coming up?
Sure. So what we see, quite frankly, is that instead of looking at those large markets as far as what population you can treat as far as patients, you start by looking at patient populations that are much smaller. And in that case, you're focusing on that where your drug works better than many other drugs that are out there. And you build upon that. So in some sense, it's a reversal where we originally started, where you only looked for very large markets to be able to go forward. Now you're looking for an area which your drug works much better than others, and then you build upon that. You see where else it can go. And that allows one to potentially show advantage of your drug early, but it also really constrains your ability to make that drug in an efficient and less costly fashion. And that alone makes it really difficult because you're actually treating folks with something that tends to be more expensive. And then you have to consider certainly the other area, we're looking at how you can even do it for fewer people, even to the point of actually developing drugs for individuals. Now that makes a very difficult aspect related to how you actually make material that meets the needs of very few individuals in the world, but in fact helps them directly. And there's a true balance there. It certainly relies on good process development, but also a good thought process of how you make that less expensively in an efficient fashion under GMPs.
And would that also then bring in disposable technology?
No question. It has actually caused that to be used. And quite frankly, that is based on what we tried and did in the hospitals starting 50s and 60s, where you went from sterilizing instruments to using bags that are irradiated, plastics, whether it's syringes or bags to deliver solutions. And that was basically scaled up in the mid-80s to early 90s to the point that it actually is useful and is certainly best to use for those small processes that need to make small amounts. That's the best and most effective way to actually make those small amounts for proteins that we have.
And I know in some of your earlier companies you are responsible for setting some of these and designing very early on these disposable facilities.
We certainly did do that. Certainly at Acceleron, we probably had the first completely focused facility on single-use equipment. And that was from the bioreactors way through the purification itself. And that actually, is an interesting and educational time because we had to help the folks that are making those bags make sure that they were integral, make sure that they didn't leak, make sure that they had significant welds that in fact survive the use in the process themselves. And I've gone on to do that certainly with a number of organizations that are looking at, well, how do you ensure that they're even better? How do you ensure particulates are not there? How do you extractables are not there?
Sure.
And ASTM is really focusing on that directly.
Absolutely. One quick question that comes to mind, Bob, is what do you think is the skill set? To many of our viewers who will be watching this broadcast will also be people coming up in the biotech space. So what would be your message? What would you believe are the new skills they would have to sort of imbibe for sort of catering to this sort of changes in CMC and process development, so on and so forth?
Right, and significant difference which has changed over the 20 or 30 years is not necessarily as focused on the chemistry to make the drugs, but actually on the biochemistry and the nature of proteins as drugs. So having a good sense of what proteins are and what causes them to have problems, what causes them to degrade is important. And it's also a question of how do those proteins function within the equipment they use for these type of processes, whether that's in the cell culture itself or in the purification. And finding ways to do that efficiently, finding ways to do that continually and continuously is certainly an area that we'd like to be able to do just to actually make the process of making those drugs much more effective and efficient. And so there, the focus is very much, what is a protein? What is its properties? What do you have to worry about when you actually go and scale that type of process up?
True, very true. So what you're saying is that it's no longer like like we don't have monotherapy for oncology. It's like when you're training scientists, you're not training them on one subject. They have to be sort of understanding biochemistry very well, protein sciences. They have to understand material sciences and so forth, along with engineering.
That's correct. It's very much a synthesis of a number of different disciplines. There's definitely an engineering aspect to it because you have to be pragmatic and practical and make it actually work. That being said, it's really helpful to have a sense of what the protein properties are,
right.
what actually, how those properties might cause it to respond differently in different equipment, and what might happen versus the conditions that you actually storing those protein or actually making those protein. And that becomes very important. And there's a synthesis between understanding what a property of that protein, what they are, and how does interact with the process itself.
Taking one step ahead, we discussed the new entrant of new drugs, personalized medicine, and then disposable technology and new skill set. That leaves another very important part behind, which is for us to discuss is the regulatory part.
Correct.
The new CMC issues. How will the regulatory bodies look at it, and what will the regulatory changes, the landscape? Your thoughts on that, Bob?
Well, it's an interesting to go through new technology, particularly with either the FDA or European authorities. But in particular, the FDA has been very focused on, can you show benefit to particular patients? And they're willing to take risks as long as you can demonstrate direct benefit. And the time that I've worked in the gene therapy field, they were quite willing to take risks related to the process and related to properties as long as the effectiveness of that drug is really substantial. And again, the risk is related to a few patients that have no other, in many cases, no other means by which they can improve. And that makes a big difference if you're able to actually help those individuals. But it is a conversation. It's very much actually, when we started and working on proteins as biologics, those were the discussions we had between 1984 and 1990, because we didn't know, but quite frankly, we were substituting for things that are extremely expensive. In fact, in a great example, and we can make it as long as it was safe for those initial individuals.
Absolutely. So the last but not the least is, as you rightly pointed out, is that the FDA or any of the regulatory agencies are going to look at the real benefit to the human patient that you're looking at. The other very important thing is bringing price down, because many of these personalized medicines as of now are significantly expensive. So I believe scale will bring down the cost perhaps, and maybe the advantage of having new technology come in.
Absolutely. The balance between making a product for a small number of individuals versus the effectiveness of that is truly the place that you need to actually look at. And it is difficult to make a product for any individual unless the process that you have can easily be duplicated. And you can easily control that which goes to that individual versus another individual. So in some sense, it's very much like parallel processing, which you potentially can actually go and approach. If the process is very similar, then in fact you can use an individual's human cells with an individual component that actually makes that T cell respond to a particular disease as opposed to a different one for another individual. But you can do that with a very similar process and move forward in a parallel fashion with a very similar process for all those individuals to be able to scale up. And to see, that's what I see actually going, is that you may have to do that in order to make such drugs applicable for those individuals that need a very specific drug.
We come to the conclusion of this talk. session, biotechnology session by Bob Steininger. Thank you very much, Bob. That was an amazing discussion. We really appreciate you coming here and for us. Thank you so much. And with that, thank you viewers for this session of biotechnology.